The most surprising was this overexpression of genes that relate to visual perception, which is kind of like, whoa, why did that come up?
Haleh Fotowat, what is a neurobot?
um, a neurobot is a kind of a biological construct that maybe you can call it a biological robot that is made out of frog cells, embryonic cells, and consists of frog skin cells plus neurons. So to kind of make it in a small word, give it a name, we called it neurobot.
Okay, talk us through. I've been obsessed with xenobots and Michael Levin for a long time now. So
Mhm.
this is why I've been so interested in this conversation. But talk us through how these neurobots were created. What is the process in the lab that you go through
Yeah.
to make these?
Okay. So you know about xenobots, right? So the xenobots are made out of frog embryos. You excise a piece of the embryo, which is the top part of it, and that part is called the animal cap in frog literature. And that's the part of the embryo that's known in future it'll become the tadpole skin. So that's how the very basic form of xenobots are made. There are different variations of that, but the simplest form of it is just cutting that part of the embryo and letting it sit in a dish for a few days, and then it will start moving around the dish. And the reason for that, as you know, is because there's a cell type that's normally also present in tadpole skin called multiciliated cells. And these cells have cilia that beat spontaneously, and those are the motors for Xenobots. So they move around the dish. So Xenobot, the simplest forms of them are basically some skin organoids, frog skin organoids, but they're special because they're motile, so they can be called robots or they can be potentially used as robots that would do something for you. Then in neurobots, the difference was so that's how I entered. I met Mike Levin, and he told me about his xenobots and all these interesting properties they have. And my background is in neuroscience, so Mike's proposal was what happens if we give neurons to this kind of completely new motile body. So imagine that you have a body, but has no neurons. But what would happen if you give neural precursors to this body? Would they differentiate into mature neurons? Would they make what kind of nervous system architecture would they make? How would having brains affect their behavior? So to make neurobots, we basically do exactly that. So we have the the basic parts for a xenobot, so the top part of the embryo that we excise, and then we implant that with some neuronal precursors that are also isolated from frog embryos. So and then this when you implant the neurons again, this kind of animal cap also heals, and after a few days they start moving around. um and yeah, so that's what we call neurobots.
Completely wild. Thank you for that breakdown. And by the way, one of the first questions I asked Michael Levin, because my background is also neuroscience, nowhere
Oh,
near
cool.
as substantive a background as your neuroscience background. I want to make that very clear. But I was also like, well, have you given it neurons? Like, are you going to give it a brain? Like,
Oh, cool.
what's going to happen when you give it a brain? Like, that was one of the first
Cool.
questions I asked him. And I think he
Okay.
said, we can't
Fire.
talk about that. Yeah, I think he was
Okay.
like, he hinted at that work is being done, and I'm guessing that was the work you were doing at the time. So
So what is your
it
neuroscience background, just so I know?
so I studied neuroscience in college, undergraduate degree. I did my thesis on the endocannabinoid system. I was
Okay.
studying how a endogenous neurosteroid called pregnenolone acts on the CB1 receptors and doing complex 1 assays. I
Mhm.
was offered to like pursue that as a PhD, but didn't end up going that direction. Ended up starting my own startup, and now
Mhm.
been running a media company for the last few years and having a lot of
Cool.
fun doing that because I get to speak to
Yeah,
people like you.
great.
So what point in the development of these uh, embryonic frog cells do you give it the neurons? Is there a critical window where you expect those neurons to become integrated into a functional nervous system? Is is there a window that you can miss where it won't become a functional integrated system? Talk about that.
This is a really interesting question. So this is not something that we have systematically investigated as to at which time window, but the what I did was, so there is a certain time window during which it's like it's called stage 9 or 10 in the frog embryo development, that if you excise the animal cap, and you have this time window to remove the animal cap in order to get the cell types that make up the frog skin. If you wait longer, then it develops further and you might get other things. So that's a certain time window for the outer parts, like for the body parts, that's is limited. But then the combination with neurons, at which point you give that to the body, is really an interesting question. What I've done so far is I've tried to match the age of the neurons to the age of the outer body. So they are kind of at the same developmental stage. And so the way it worked, this is also very interesting. So this top part of the frog embryo is kind of like, where it has interesting properties. It's kind of pluripotent. Like you can make different cell types out of it. Like if you just keep them, excise them, they become skin cells. If you give them different growth factors, they might become neurons or heart cells or different things. And one thing that was known before was that if you excise that very same piece that would become frog skin, if you put it intact in a dish, and you dissociate the cells in that piece of tissue so they're not next to each other anymore, then and you wait a few hours, then those cells would become destined to become neurons instead of skin cells. So that basically the default mode of these cells is to become neurons, but when they're in a tight piece of tissue, there is inhibition across cells that inhibits the neural fate and makes them become skin. But if you separate them, that inhibition goes away, so then they become neurons. So the way that I did that, I collected embryos that were fertilized at different times. And for the outer, so I first took this animal cap, um, dissociated the cells, waited for a few hours to make sure they would become neurons. And then I reaggregated those cells, isolated the animal cap from a different embryo, which was fertilized 3 hours later. So to kind of match the developmental stage of this outer part and the inner part, and then went from there, and that worked. But it's very interesting to look at that to see, okay, if we took that and then put older neurons in there, or if we put, neurons
Right.
later, it's already moving, because at the point where these precursors were implanted, nothing, like it's just the very beginning, it's not moving. It still takes a couple of days before the whole thing moves. So they all develop together. But your question is very interesting, something to look
Very
at.
nice. What the listener at home might be thinking, like, what is the goal of this kind of research? So we're taking
Mhm.
these biological systems and we're taking away the usual constraints that those cells are under. And we're just
Yeah.
we're kind of shrugging our shoulders and saying, well, what's going to happen? Because we don't know, right? We've never
Right.
taken this biological system out of its usual constraints. All of these
Mhm.
different stimulus that usually constrains the system in all of these ways, and we don't really know
Yes.
what's going to happen. So what is the
Mhm.
goal of this kind of research? What do we do, or what's our hypothesis going in? What exactly are we trying to study when we do this?
Yeah. So there are, so at the very basic level, what's really interests me a lot is to understand um, the rules that cells follow, the rules of self organization, and how adaptable our biological cells to new situations that they've never experienced before. So it's a very basic question that we're trying to address. And then, from there, we would learn a lot that would eventually have applications in a lot of different areas. So, for example, an example of cells being in a situation in which they've never been before, you could think about in the context of an injury or, in the body when the situation is different. So this could potentially in future have applications for, um, you know, what do you call it, regenerative medicine, that you would say, okay, this is the situation. We know the cell type has these kinds of properties, and we have found uh, the knobs or the points at which we can, manipulate the cell to do what we want. Because here, like, we are not, to start with, in neurobots, we didn't do any kind of engineering and manipulation. We're just right now watching and see what they do normally on their own intrinsically, what are the tendencies. But then once we learn what those tendencies are, then maybe we could push more in that direction and get them do more of that. So
Right.
that's one aspect of just learning how biological cells behave in new contexts. The other thing is, the obvious from the name is, if you wanted to build robots out of living cells, we need to understand how those cells behave. And these could have a lot of different applications. For example, all the things that you would think robots these days do that are useful for us. If you could build them out of living cells, it would give you a lot of different types of advantages. For example, they could repair themselves, they would biodegrade, they're adapting to their environment and all of those things, that they could find applications in places where you can put like a rigid electronic robot in, or they would work better. I mean, each of them might have advantages in different situations, but I think personally find building machines with living cells very exciting.
It's very cool. I was just yesterday I was speaking to Brett Kagan. He's the chief scientific officer of Cortical Labs. And I was
Yeah.
really trying to understand his vision for the future of this.
Mhm.
So recently the headline went viral that they got a bunch of neurons in a dish to play Doom. It actually wasn't even them. It was just a guy that was piggybacking on their sort of
It's
experimental apparatus, which I find is
yeah,
fascinating. The thing that went viral
yeah.
wasn't even the main team. But he was talking about a future where, instead of me doing some sort of computation on my laptop and it's pinging some giant server somewhere, I'm actually
Yeah,
pinging into a server farm of neurons somewhere. And
right,
those neurons are actually better at doing some sort of computation than silicon are.
right.
And that's bizarre. That's really weird. Like
Yeah,
that's a really strange idea, I think. Maybe you've spent a lot of time thinking about this and it's not so strange to you, but the idea that I'm on my phone or something and
yeah,
I'm doing something and I'm sending a signal and that computation is being done by actual neurons somewhere, and then the output
yeah,
of those neurons is being sent back to my phone. Like that's
yeah.
wild. That's a weird.
That is pretty wild, and there is a lot of also, uh, conversations about energy efficiency, and if you can have, like, brains are so much more energy efficient than our computers, and
Yes.
how can we make that? And yeah, I think a lot of those biological computation kind of ideas is like that. They're like, okay, you save a lot of power, maybe. I don't know. But I feel like it's very exciting. We're not there yet, but I feel like, yeah, we're kind of
Does
inching towards that.
does the idea of having like a giant warehouse full of human neurons, it kind of weirds you out a little. I think is that like a bit of a when I think about that, I'm like, um, maybe I have no idea. That sounds weird. I'm like,
Oh,
energy
it's really
efficiency for
cool.
sure, but I'm like, that's like we've just gone out like. We have to at least, have a conversation on how weird that is, even if it is more energy efficient.
It's interesting. I don't know. It somehow doesn't really weird me out, but maybe it's because I've been
Okay.
cells all the time, but I find that really cool. But it's true that it's like, yeah, you think that's something squishy that like you have to feed it like actual sugar and for it to do work. Yeah, it is weird, but
Someone has to go feed the warehouse on a Monday morning.
yeah.
So
Yeah.
so let's dive into a bit of the results that you saw. What did happen to the clump of frog skin cells when you gave it a, I'm not going to say brain, when you gave it a very primitive nervous system? Did the neurons actually gain that functional control over the skin cells? Did it not? Talk about what you actually observed.
Yeah. So for one thing, yeah. So one thing is that the neurons did grow because that was not a given either, that they might just die and not differentiate into neurons. They did differentiate into, mature neurons. We showed that they make, then there's this question, do they make all these different like axons and dendrites and synapses and all these things? And they do. So they do have both axons, dendrites, synapses. And um, one of the big questions that I had was where are they gonna, what kind of structure does nervous system would make, which we still don't have a real quantification of that. Because, for example, when we were talking to Mike Levin, the question is, if you look across all the animal kingdom from the very simple nervous systems to really complex nervous systems, will you get something that more looks like a nerve net that you see, in Hydra, for example, or would it look more like an insect brain where you have all these different nuclei? And what would happen? But I would say, like, we still don't have an answer to that, but it's something simple, simpler than those. So their way, the way that these neurobots are made, they were all done by hand, by my hand under the microscope manually. So each of them is so different from the other one. So if you look at the structure of the way the neurons have grown, there is no like, oh, they all look the same, and that kind of is expected. But there were some things that were common across all of them, which is they kind of course, I have this clump of neurons, so they start growing out of that clump. And so that it looks like it's like a nucleus of cell bodies, and then all these neurites are coming out of it. But that's the result of the starting point. It's possible. I mean, it's also true that it could have been that after I put the clump, it would just fall apart and do something else. But it didn't do that. It looks like the original clump that was implanted, the neurons, the cell bodies start from growing from there and then sending out their neurites. And then they kind of look like they're making this network amongst themselves. But in all of the neurobots, we also observe that there are these processes that go towards the outside of the neurobot, which is the part that was exciting because these are, you have to travel somewhere. It's not far, but the fact that they kind of grow towards this other cell type, because they're all neurons in the middle, they're implanted, but then, I mean, I don't know, that there might be also astrocytes and glia. We haven't looked at that, but the outer cells were what we were interested in, because if the neurons are to change the behavior in any way, they have to communicate with the motor cells that are on the outside, some either directly or indirectly somehow, but they have to, like, the best way is to kind of make it there, to get close to them, to be able to talk to them. And they do. So in all of them, we see this projection towards the outer cells and anatomically. And there are some interesting changes in morphology also that comes out of neurons growing in there. With the non-neurobots or xenobots or biobots, they're more circular, and the multiciliated cells that are the motors are more uniformly distributed on the body. Whereas for neurobots, the distribution of multiciliated cells change. It's in interesting ways. They become more asymmetrical. The whole body becomes more elongated. And we showed that the elongation of the body is correlated with the degree of neural growth inside the neurobots. So it could be, we don't know what causes what, but somehow this axon growth affects the mechanics of growth of the other cell types. And then, yes, these were some of the structural findings that we show. And then we also look at behavior to see if the behavior of the ones that have neurons is very different from the ones that don't have neurons. And like, if I give you a microscope and I kind of ask you, okay, which one do you think is a neurobot and which one do you think is a xenobot? You probably couldn't tell by just looking at their spontaneous movement. They're very similar. You could more tell by their shape because, as I told you, neurobots tend to be more elongated and have like a different pattern of distribution. But overall, you don't see that, oh, all of a sudden their movement is completely, absolutely different. There are some subtle changes in movement. So one thing is that neurobots in general tend to be more active. So if you look at the percentage of the bots that have neurons that move around a lot versus the ones that don't, neurobots are more likely to be active. Like, I forgot the numbers, but that was a significant difference between the two in the level of activity. Spontaneous. So these are all spontaneous. And then the other thing is that overall, the complexity of the trajectory seems to be more complex, spontaneous trajectories. Um, so in the paper, I have some of the examples of the trajectories. So it's interesting because you look at the movements. Some of them like to go around a circle. Some of them do these kind of cool spirograph patterns. And then some of them do various things. I really like watching those trajectories a lot. And in general, like you see that the non-neurobots are more likely to do circles, just simple circles, but then neurobots might have more complex trajectories. One thing that we are now working on to see is whether or not having this complex trajectory uh, how much of it can be explained by the difference in the shape and the distribution of multiciliated cells, because that's also different, and how much of it can be explained by neurons modulating the way that the biobot moves. So we don't have complete answer to that yet. But we have shown that neurons are definitely active. So doing calcium imaging, they're spontaneously active. And also when we treated the neurobots and non-neurobots with this drug that induces seizures in animals, we see a significant difference in the way that they behave. So we think that probably neurons are doing something in modulating the movement of the bot. But how are they doing that? What is the mechanism? What is the extent to which they matter? These are questions that we have to address. Also, another thing that we don't know so far, we've only been looking at spontaneous movements. We haven't really explored responses to stimuli. So it could be that the spontaneous movement is not that different, but then all of a sudden, if you show stimuli, one might respond more than the other or differently. So these are all still open questions. And the other thing is, this is just a start, right? Like you would anyway, I would go there. Maybe you have more questions. I stopped talking.
Talk about the altered gene expression of these neurobots, because this was something that
Mm,
was super interesting to me. And again, you kind of read it and you're like, I don't know how to interpret that.
yeah.
I don't know what I should get from that result. So talk
Mm.
about what happened when you did the gene analysis in these neurobots.
Yeah, that was really interesting. So we did this gene expression analysis to compare three different types of bots. So there were neurobots, and then xenobots or biobots is what we call in the paper, and then non neurobots. Is that what I called them in the end? Yeah. non-neurobots. It's hard to like give terms to these. And the non-neurobots were made the same way as neurobots, implanted by a clump of cells. So to kind of keep that kind of mechanical action the same as neurobots. But then, as I told you, um, the neurobots had to separate the cells, dissociate and separate, and wait for a while for them to have the neural fate. For the non-neural version, we didn't wait the 3 hours to kind of dissociate and put them back immediately together, so they don't become neurons, but they're just an implanted clump. And then compared the gene expression between these three different categories. And neurobots were different from both. So the non-neurobots and biobots were very similar to each other. So they're basically all skin cells and didn't have much neurons. Biobots had nothing. Non-neurobots, some of them, very small portion of them, had some neural gene expression, but not nearly as much as the neurobots did. And so the part that was expected was to get an overexpression of genes that relate to nervous system development. And that's what we got in neurobots compared to biobots. So that wasn't really surprising. But then some of the other surprising things were, in terms of, to me, the most surprising was this overexpression of genes that relate to visual perception, which is kind of like, whoa, why did that come up so
Exactly.
much? That was really interesting, especially since it seemed to be really. So I learned something, that maybe this is how the self-organization works, is to like, you don't only upregulate genes that make up photoreceptors. Once that happens, it seems like everything that relates to that goes together, which was interesting. So it's kind of, we get upregulation of genes that make proteins that you use to make the lens. So the lens part, photoreceptors, rods and cones, all these different types of retinal cells, all of the stages, at least from the eye part, were there. And these are all, I think it's important to note that they are all just at the level of RNA. So we don't know if they were actually made into proteins. So this is something that we should look into to look at the protein levels of rods and cones as opposed to the genes, because the genes might be upregulated, but they're not actually there, or maybe they're not there yet. So this is something that we have to look into. But it was super interesting to see um, that result. And yeah, so the next step is to see, are they actually there protein wise, and are they functional? Are they doing anything
Like,
functionally?
yeah, it's just so I like when I it's the kind of thing that I read and my brain just goes in like 40 directions. Like one sort of naive interpretation of that is like this system is just like dropped into a new universe that it doesn't understand because all of its usual constraints just aren't there, or it's a different pattern of constraints. And then it's like, well, what do we need? We need a sensory system. It's like it's trying to figure out a way to navigate its world. So let's build a sensory system. Like let's
Yeah.
put together the building blocks of that. But then that is like
Yeah.
seriously applying a level of agency and a level of
Yes.
almost sentience to the system where it's like able to figure that out, or able to, like, why does it just reach, like, the genome in those cells is so expansive, right? The amount of things that it could reach for um is massive. So
Mhm.
why is it throwing darts at a dartboard that just happen to be vision because of the exact strange pattern of these cells? Does this happen all the time? Does it always upregulate visual genes regardless
Yeah.
of the specific neural architecture? It does.
No, I don't know. So this is a very,
Okay.
very good question. So first of all, these were done, the RNA sequencing was not done so there are groups of them. So I can't know at an individual level, because the amount of tissue was so little and the methods that were used, we needed more than one neurobot per tube to do the RNA sequencing. So there were like 4 or 5 neurobots per tube. So we don't know individually if they're different, but this is something we should look into in future. The other question that you asked was, yeah, were all of them like that? That's a very good question. We don't know. But I think now the methods are a lot better. So we can, moving forward, maybe also we can move into proteomics to see is the proteins are the proteins expressed, and then look at individuals, and then also look at behavior of those individuals, see if they respond to light or not, and if that correlates with the expression of those. But why do they pick vision? Yeah, it might be just totally random. I don't know. Like it could be that in the sequence of development of sensory organs, vision is what comes up around that time. It
Right.
could be totally random. It could have to do maybe with environmental conditions. For example, one interesting question to ask is if you change the pattern of visual stimulation, would you get a different um, expression level? Or if you expose them to other types of sensory stimuli, would that affect anything? I don't know. I think these are all really good questions. And yeah, I think sometimes I think if I was gonna do another PhD or master's or whatnot, I would do it in developmental biology and developmental neuroscience, because maybe some of these questions are trivial you're asking, but I don't know. I think these are all very, very good questions.
Because the xenobots upregulated genes for hearing, right? So again, it's really easy to like first analysis kind of guess what these are doing, but it's like it really feels like it's just trying to grab a way to navigate its environment. It's like, what are we doing here? We don't know what we're doing. Pull at the nearest sensory apparatus that we might be able to build so that as a system, we become better at navigating this strange terrain that we're in. And it's just starting that
Yeah.
system and reaching for the thing that's most likely. But that's also very
Yeah.
much treating the whole organism like it's all fully one thing. And it's very, we don't really know how you did some really cool experiments to show that there is nervous system regulation going on, but we don't know how to think about this clump of cells as one thing or as a clump of cells. All these questions make my brain sort of melt and tie into a croissant. That's why I think they're fun to talk about. But
Yeah.
like, why does it seem to be reaching for a sensory system? It feels like it's trying to get better at navigate its environment. It's like we need to survive here, guys. We need to get our shit together here. Like we need, it's like it's arming itself with equipment.
It could be that. It could also be that this is, the and as these cells develop normally in a tadpole, some of these cells would become retinal cells, and some of these become whatever they want to become. And that they continue doing that, just kind of the same way as the outer body of the biobot. Those are skin cells, and they become skin cells. It's not like they were reaching for something. They kind of, again, goes back to this agency question. It's not like they're like, oh well, what do I do? I have to sense the environment. So it could be that in terms of the developmental process, this is kind of what they would become. And then what really interests me to a little bit study more is, and this seems to be very important, is environmental constraints. Like you are now inside this mechanically, all these mechanical forces that are different. Um, that you're constrained by that, and there's this thing that's moving. So how does that affect the fate of itself? Like what would that become? How is that affected? Like this is known that it's a known fact that cells are affected by their environment. Like pluripotent cells would become different things if you put them under sheer stress, or to give
Right.
them electricity, or all these things that are happening in neurobots in a different way that we don't fully understand mechanical forces in these cells. Maybe that's something that results in getting this. Uh, I think more of it that way than
Yeah.
um, well, let's see how we can sense out of the environment. But again, like you can say it differently, and that define that in that
Exactly.
way.
I'm like semi joking, but I'm only semi joking because we just don't really know. How long can we
Yes.
keep these systems alive? Like it would be so fun to think about, like, is there a way that it could hack together its own photo
Yeah,
receiving organ? Like, could it hack together the pieces in the right order
yeah.
to actually get some sort of photo receiving signal if you give it enough time and enough iteration cycles?
I think that those are the experiments that I'm planning to do. We tried it. You can keep them alive for a while. So they're they don't need to be fed up until 10 or 12 days after they're made. They have their own individual cells have their own yolk content. So if you don't after 10 days, if you don't feed them like you didn't before, then they would die. They would disintegrate and die. But you can feed them. So that means that now you would have a different setup. So one thing that's cool about the xenopus cells is because they have their own food source and you don't need to feed them the first few days, they don't need sterile conditions. So you can just have them in a dish of media, which is mainly like salt water, and they swim around and nothing gets infected. And like, unlike, for example, brain organoids or other organoids that are made out of mammalians, have to be in sterile condition all the time because you need to feed them. These then they don't need that. But then if you want to keep them longer, then you need to feed them. Then they have to be in a sterile condition. And people have done this where you can keep them alive for months and months. So that would be interesting to look and see. Another fun fact about them is that I forgot after how long, after a few weeks, they start losing their multiciliated cells, and that's what happens also in their normal development of tadpoles. So initially, tadpoles have a lot of these multiciliated cells because they help them glide around a dish and remove debris or other stuff there they don't want. But that's before they start actively swimming. Is that they're gliding using these multiciliary cells, but then after a while, their muscles develop and they start actually swimming, and then eventually they'll lose those multiciliary cells. So in the case
Interesting.
of xenobots, neurobots, it means that after a while, you'll have less and less of those ciliated cells, and you would move less and less. So depending on what you want to do with them, there might be a way. Then, I mean, you could always kind of have a different version, genetically modified version to keep expressing those. But if you're using the normal unaltered cells, then after a month, then you probably are not going to be moving anymore. But it would be still really interesting to see.
The Giant Shoulder mission is to explore radical ideas in biology, neuroscience, and consciousness, and elevate those stories to the highest possible level while keeping them accessible to everyone. If this interests you and you want to support independent science, then please consider subscribing to the Clips channel